Okay, so welcome back, everyone. My name is Ted Fawn. For those of you who don't know me, I'm here at the Neuro. I'm the scientific director, and I also work on Parkinson's disease. Although I'm not a hardcore GBA researcher, so many of you probably don't know me. And I have to say, I'm really thrilled to have this meeting here at the Neuro, and I find that it's already, just in the first session, I've learned so much, and I think this is the kind of meeting really focused and diving deeply into a topic like this, which is so important for Parkinson's disease. I want to congratulate Ziv and the team that put this together. So not going to take more of your time, and I'm going to introduce the speakers of this session on the mechanisms of GBA-associated neurodegeneration, and remind the speakers that each talk the format's going to be a little different. There's not going to be a session at the end. So after each talk, we'll try to have 5 minutes for questions. So each talk should be 20 minutes or so we can have that time, because I think the questions are particularly informative and useful. So to get the ball rolling, Joe Mazzulli from Northwestern University is going to start. And Joe, of course, really one of the pioneers in connecting GBA and synuclein. And he's going to tell us a little bit about lysosomal dysfunction and synuclein accumulation. Joe.
Great, thank you. It's a pleasure to be here. I'd like to thank the organizers for the invitation. So, disclosures. Okay, so I'm going to talk a little bit more about synuclein. That's the main focus of my lab, synuclein aggregation, and how those aggregates lead to neurotoxicity downstream. And so, you know, the reason why we study synuclein, we study connections between synuclein trafficking and lysosomes, and this is really the main reason, because genetics has identified not only GBA1, but many other lysosomal hydrolases, channel proteins, and other proteins in the trafficking pathway. Here we go. That all sort of, we think, converge. And on the right here are some of the, on the left are some of the key studies that identified some of these variants. Okay. And so when you look at what synuclein does in terms of its toxicity downstream, there's really a strong overlap between the genetics and the pathology here, as I have shown in this slide. And the studies that we've done over the last 10 years or so have identified when you overexpress synuclein or it accumulates in some way, and a lot of our studies have been done using patient-derived iPSC midbrain neurons that have mutations in SNCA that induce its aggregation. And so a lot of these variants are likely to be in the wild-type form, and so we're only studying the effects of when synuclein is building up. We've been able to show that there's actually problems in the nucleus, which I'll tell you a little bit about later if there's time. There's hydrolase misfolding and accumulation in the endoplasmic reticulum. For example, glucose cerebrosidase accumulates in the ER, and synuclein causes this dramatic fragmentation of the ER. It disrupts vesicle fusion. Our lab and a few others have studied this, and we think it's by impeding specific snare proteins that mediate vesicle fusion. And what I'm going to focus most of the time is this last part here on how we think this whole process initiates in the context of a GBA1 mutation. And that's through the connection with glycosphingolipids. So just to give you a brief background on synuclein, physiologically, it's found in synapses. It's a presynaptic protein. This is an immunostain of a hippocampal primary culture from mouse, showing that it co-localizes with a presynaptic marker. It binds preferentially to acidic phospholipids. I think we heard some of this in some of the earlier talks. It associates with lipid rafts in vivo, and this is, I think, important for some of the toxic mechanisms in terms of GBA, Parkinson's disease. And we know that it aids in vesicle snare proteins in somewhat of a way of maybe like a chaperone-like activity, where it promotes vesicle fusion at the presynaptic terminal. You can see this is an immuno-EM for synuclein, showing that it's concentrated in presynapses. So some of the basic properties of synuclein as a protein: it's a natively unfolded protein, it's heat stable, and it's has three main parts. The N-terminus has this lipid binding domain, which is very similar to exchangeable apolipo binding proteins. This is the area where a lot of the mutations that cause Parkinson's disease are found, and some of the point mutations are listed here. And another thing that I'm missing is, of course, triplication of the duplication and triplication of the SNCA genomic region is also linked to familial PD. So the middle part of the protein is called the so-called NAC region. It's a region that contains mostly hydrophobic amino acids, and we think this is the region that is required for forming these amyloid aggregates that you find in Lewy body inclusions in Parkinson's brain. The C-terminus is highly negatively charged. It's an acidic region. It's highly mobile, has chaperone-like properties, and also has been shown to bind catechol, which may be some connection, we think, some connection to the dopaminergic cell death that synuclein is involved in. So under the physiological state, I think synuclein is a really interesting protein because it can assume different structures. This was an early paper by Peter, which I think was probably one of the first to characterize its structure when it was purified to homogeneity from E. coli, showing it's an unfolded monomer, but can also sample different conformations. It depends on the environment. For example, when it's in the presence of phospholipids, it can assume this alpha helical structure. Some groups have shown that it can form tetramers, and some of the work that we have done has shown that it can form higher structures like multimers, and these all occur under physiological conditions. So in the context of GBA1, of course, we're always interested in how these variants lead to synuclein aggregation and conversion of those physiological forms into the aggregated forms. And there's two main hypotheses that we're going to talk about later this afternoon. But just to briefly mention, we have evidence for both of these. In terms of the ER stress, this is the gain in toxic function in terms of how these mutations cause ER stress and how could they lead to synuclein aggregation. We don't really have a clear explanation for how, because this is such a general type stress, how that could lead to specifically synuclein aggregation and Parkinson's disease as opposed to other diseases like Alzheimer's disease. Loss of function mechanism, there is a little more there to explain how there could be a specific connection, at least in my view, where you have a reduced in lysosomal GC activity and accumulation of glycolipids, and synuclein can interact with those glycolipids based on its lipid binding properties. So the model that we mainly use are patient-derived cells from Gaucher's disease or Parkinson's disease patients, and these are some that I told you about before with SNCA mutations, so they'll accumulate synuclein, or some that have heterozygote GBA1 mutations. This process takes about, once you have stem cells, it takes about 60 to 90 days to differentiate into midbrain type dopamine neurons. And this is characterization of some of those. We do a lot of quality control before we use these cells in experiments. We can see that after about 60 days that colocalization of synuclein with some of these presynaptic markers occurs, indicating that these are likely mature neurons, and we think it relevant to the adult stage. And there's many other midbrain markers that we use to characterize these cultures, and they're relatively homogeneous. They're not pure, but I would say 90% neurons and probably about 80% tyrosine hydroxylase positive. So using these cells, this is an example of some of the ER stress phenotype that we can detect in GBA1 mutant cells. These are Gaucher's patients that have the N370S and 84GG mutation. One of the things that happens when the cell is trying to refold a misfolded protein, like for example N370S, is the ER will expand to accommodate that extra load of protein and chaperones that are upregulated, for example, to try to fix this problem. And so you can see that actually occurring in the GBA1 patient cells, where you see this dramatic expansion of the endoplasmic reticulum. We can also see protein levels that are associated with the unfolded protein response, like GRIP78 and EDEM that are going up with either the L4P or the N370S Gaucher's disease midbrain neurons. And we're not the only one that showed this. There's other people that have shown that as well. So in terms of loss of function, this is really the reason why we've been focusing more on loss of function, is because of lessons from Gaucher's disease. We know that the mutation causes a depletion of the protein. It degrades it through ERAD. There's a dramatic reduction in activity, and we can see this is total glucosylceramides going up in these cells. This is an example of an EM image of a control cell, and you can see in the Gaucher's situation, there's lots of accumulation of vesicles. This is a little hard to see, but it's a multivesicular body. There's electron-dense vesicles, there's electron-lucent vesicles, and multilamellar bodies. So lots of stuff is accumulating in these cultures. And again, this takes about 60 to 90 days to achieve that type of accumulation in vitro. We can also see α-Synuclein accumulating in the insoluble fraction. So this is really the insoluble portion of this is what we think corresponds to the amyloidogenic or toxic forms of synuclein. This is a Western blot showing using different forms of, or different antibodies against synuclein, showing using either for total synuclein. This one was generated against Lewy bodies. This one, 303, reacts against oxidized and nitrated synuclein. So that's a pathogenic subtype of it. But using all three, we can see a clear indication that synuclein is accumulating in these cells. And in fixed cells, we can actually see juxtanuclear inclusion bodies that are thioflavin S positive, indicating that they have an amyloid-like conformation. This is happening in cell bodies and also in neurites. So we think this is a pretty good model in terms of recapitulating several of the important features of disease. So one of the reasons why we think that loss of function is playing a big role here is because we can actually take wild-type cells, these are healthy control iPS midbrain neurons, and add the inhibitor conduritol beta epoxide. And after 2 to 7 days, in that range, we can induce, these are cells that express wild-type glucose cerebrosidase. We can, just by reducing the activity, cause the accumulation of synuclein. It looks very similar to what we see in the Gaucher's patient cells. See about a 2-fold increase and an accumulation of thioflavin positive synuclein. Where does this happen? Well, we think a lot of the action is happening in lysosomes. We could see a co-localization. These are primary cultures where we knock down GBA enzyme or GC enzyme. And we can see a co-localization of synuclein with this lysosomal marker, LAMP1. We can also see a co-localization of synuclein with the glucer antibody. And in vitro, we can actually get, when we mix purified lipids, glucer lipids, with purified synuclein, we can see that it enhances the formation of amyloid aggregates. So we do think this is a direct effect. This is an EM showing synuclein fibril here, which is a little hard to see. And here are glucer tubules, which is something that happens when glucer accumulates. It forms these gel-like tubules. Okay, so getting back to this question of what structure is involved in initiating this whole thing. And again, because synuclein is sampling all these different conformations in the physiological state, we do think that's one of the things that makes it susceptible to going down that pathway of amyloid aggregation. And so one of the things that we found in our midbrain cultures was that we can detect high molecular weight oligomeric species. We can also detect monomeric species. And so we thought that maybe when lipids accumulate, they may break down these oligomers, the physiological oligomer, and into its monomeric components, which then form amyloidogenic aggregates. This is normally the way protein aggregation occurs. You need to break down physiological structure. But we found actually the opposite. We were a little bit surprised. What we did was we isolated different fractions of synuclein and then mixed them with purified glucer, or using this control lipid phosphatidylcholine, and using a FRET assay, which is a way that you can look at protein structure sort of indirectly. We couldn't look at it directly because we couldn't purify the protein to in a way that could get good quality structural or biophysical data. So we had to use an immunoassay for this. But you can see when these antibodies, these two of the same antibody that have different fluorophores, and when they come together, you get an increased FRET signal here. And so it sort of works like what I have shown here. When you add physiological oligomers to glucer, you can see this dramatic increase in the FRET signal, indicating that something like this structure is occurring, where it's a more compact structure. We did see similar things with monomer. It wasn't as dramatic, though. This is the black line here is the monomer. And so what this indicated was that really what was happening, glucer is directly converting the physiological form of synuclein, the high molecular weight form, we think, into this assembly state oligomer, which then goes on to form fibrils. We published a lot of this work a while ago in 2018, and this is the pathway that we came up with. This is the way we think that it occurs. And so the next thing that we studied was if we can reduce lipids in iPSC cultures, these are Gaucher's disease cultures, can we reverse all this? And we were able to get a nice rescue using the glucose or the GCSI inhibitor, L-glustat, treating cultures for about a week or two. I think this is 2 weeks actually. We could see a reduction in the insoluble forms of synuclein and a reduction in the thioflavin S positive aggregates in the Gaucher just by inhibiting the synthesis of the lipid alone. So in vitro, that works very well. So how does synuclein go away? Well, we think that when lipids go down in Gaucher cells, it gives them an opportunity for other enzymes like cathepsins to work and get rid of it. And so in this experiment, what we did was we inhibited lysosomes. This is lupeptin, which will inhibit many lysosomes. We used a couple of other inhibitors for other pathways, like the proteasome inhibiting epoximycin and macroautophagy. And really the only change that we saw when we could reverse this effect was with lupeptin. So in a little more detail, we looked at cathepsin inhibitors, cathepsin B inhibitors, and they're not specific for cathepsin B. I think they inhibit cathepsin B and cathepsin L, but we were able to see similar effects when those cathepsins were inhibited. And so what this indicates that how we think this is happening is when we reduce glycolipids in a Gaucher's culture, it actually reverses the structure of the pathogenic oligomer into a physiological one that is more susceptible to clearance by these lysosomal cathepsins. And that's how we think the clearance of synuclein occurs in those conditions. Okay, so I'm running a little bit low on time. I'm just going to quickly go through this. We were able to see very similar things in vivo. We used the CBE mouse model where we inject mice with CBE for a week, and then followed by that, the GCSI inhibitor, venglustat. Under these conditions, we were able to see the activity of GC is low, both in the conditions of CBE, as expected, and when we have the GCS inhibitor. So activity is not changing here, but synuclein is. And here's the lipid data. This is total lipid. And you can see we could, under these conditions, find a reduction in pathogenic forms of synuclein. This is an oligomer assay where we can see reduction in pathogenic oligomers. Okay, so Peter talked a little bit about this before, then probably in a better way. I think there's not one glucer, there's many different types of them. There's a family, and the neurons, I think these are the main ones. They actually go down to C16. They vary by the fatty acyl chain length. And what they do is when they're inserted into membranes, they cause more rigidity, and they form these gel-like lipid rafts. They're generated by different ceramide synthases in the endoplasmic reticulum. That's where we think where they come from. So we looked at these different isoforms just to draw some correlations between what we were seeing in our mouse rescue model and with synuclein. The first thing we noticed is that venglustat really does a better job at reducing those longer chain lipids, C22 and C24. These shorter chains were trending downwards, some of them, but not really significant. We did a correlation of the chain length, fatty acid chain length, with synuclein levels, and so what you can see here is a correlation of, for example, C24.1 glucer with insoluble synuclein. And these are all the mice from the CBE plus venglustat treated group, and there's some variation within that group. And so we're seeing this trend, so the mice with the lower levels of the C24 also had the lowest levels of synuclein, and the same trend was true for C26. So we think these longer chain length species are actually more important for triggering synuclein aggregation. We were able to directly show this using wild type human midbrain neurons, what we did was we treated those cultures with either C16 liposomes, Glucer with the C16 chain length or the C24, and really only the C24 variant was the one that was able to trigger insoluble synuclein directly in vitro. So we think this is sort of where what could be happening in the cell. We haven't done detailed cell biological studies, but we think that, for example, as Glucer is concentrated perhaps in lipid rafts, its internalization through the endocytic system where it ends up in lysosomes. This is where we think it meets synuclein. This is where we think the action is happening. We know that we need acidic pH for this to occur. And so one of the things that may be happening, this is a hypothesis, is that Glucer may be redistributing in addition to accumulating. And we think you only need a subtle accumulation for this to occur. And this may form somewhat of a scaffold or a template for to trigger the aggregation of the oligomer into the pathogenic form. And we have some evidence for that. These are the gel-like tubes that Glucer forms in vitro. This is an immuno-EM for synuclein. When we mix the two together, we can see that they're sticking and sort of forming this template for polymerization. But more studies are needed to look at that. Okay, so in just the last minute here, I want to tell you about some of the downstream effects that we have looked at. Most of these studies are, so I told you a little bit about how we think synuclein aggregation is initiating. Now what happens downstream of that? We've looked at patients with SNCA pathogenic variants, mainly the A53T, which promotes aggregation, synuclein triplication line, and these have wild-type GBA1. So one of the things we found was that in the nucleus, which isn't really a largely studied thing in synucleinopathies, we did an unbiased proteomic screen and found that these RNA binding proteins, for example, were accumulating in the nucleus, causing changes to RNA editing, and in addition, feeding more into those aggregates. And this is an example of some of those inclusions that we can see in dementia with Lewy body brain. This is a stain for no-no and poly-A mRNA. So we think RNAs are actually accumulating in the nucleus. We just published this recently, a couple of months ago in Neuron. And that was through an unbiased approach, completely unexpected finding. When you go down to the ER, you could see that synuclein is causing this fragmentation of the endoplasmic reticulum. It's causing glucose cerebrosidase to aggregate into these insoluble fractions. This is the synuclein triplication, a Western blot compared to the control. And this is a super-res confocal showing this fragmentation of the endoplasmic reticulum. So we think this could be severely compromising the folding pathway. We also published a paper showing that N-glycans are perturbed as a consequence of synuclein accumulation, and this could indirectly affect the folding of glucose cerebrosidase. When you rescue this pathway, we can improve the folding of glucose cerebrosidase and rescue lysosomal activity. That was a paper published last month. And we also know that vesicle fusion is perturbed by synuclein by interacting with these snare proteins. And we're currently working on combination methods of really drugging the ER, protein folding in the ER, as shown here, for example, by diltiazem or other compounds, enhancing trafficking by farnesyltransferase inhibitors. We're doing some of that work in collaboration with Peter. And we think drugging both of these pathways as opposed to one single may have a synergistic effect and be more beneficial in rescuing lysosomal activity where you have synuclein aggregating. Okay, so I'll end there. I'd like to thank my lab, former lab members, collaborators, and funding from the NIH, Michael J. Fox, Cure PD, and the Toffler Foundation. Thank you.
Okay, so we have time for a few questions, so you can maybe step up to the microphone or just raise your hand. There's someone, yes, Abid.
Hi, Abid Usleti from Université Laval, Quebec City. Thank you for the presentation. I'm just curious to know if you have looked at the phosphorylation level of alpha-synuclein in your model, since phosphorylation is known to be a marker for aggregation and also affecting the interaction with membranes and vesicles.
Yeah, we haven't really looked at that. I think that's a good question, especially in terms of how is it influencing membrane interaction. But a lot of people use that assay to identify pathological forms. We've been able to do it with things like thioflavin S and the antibodies that we use. So we haven't got into 129 studies.
Yeah, hey. Can you talk a little bit more about cathepsin B and the...
Yeah, so cathepsin, we think that, I could go back to that slide here. Here's the cathepsin slide. So we think that maybe this would be a little easier to explain it. We think that in order for a drug like venglustat to work, you need to have functional cathepsins in the lysosome in order. That's what our studies are showing, is that when you make them dysfunctional, synuclein is not being cleared. And so I guess there's a possibility where you could have a cell that maybe there's an additional variant. We have a GBA variant and a cathepsin variant together. And if you would to give a drug like venglustat, you may not see that beneficial effect. Reducing lipids alone is not going to do it in that case. Because it's cleaving, we think it's directly cleaving synuclein. It's this state of it is the form that's being degraded in the lysosome.
And how does that connect to the length of the carbon chain?
So that would be before cathepsin. We think that the long chain glucers that are driving this process in the right direction, towards the right, that's what the long chain lipids are doing. When you take those down, you now convert the structural studies that we've done indicate that you're going from this assembly state compact structure into this relaxed structure. And we don't think cathepsins they can probably degrade these as well, the assembly state as well. But this structure we have found is much more susceptible to proteolytic degradation.
So 2 more questions up there at the microphone.
A really nice talk. We know other lipases are genetically associated to PD in terms of the single lipid catabolism pathway. I'm curious, do you think this is a specific property of GK substrates, or do you think accumulation of galliceres and gangliosides would also cause these kind of similar changes in the membrane fluidity and interactions with synuclein?
Yeah, that's a good question. We've studied a little bit of, just very briefly, some other lipids, but I know other groups have shown that galactosyl ceramide, for example, can do similar things. So we think that it could be the same type of mechanism may apply to many different sphingolipid storage diseases.
Okay, last question, Roy.
Excellent talk, and thank you for advancing the field about the link between GKs, the lipids and the alpha-synuclein. Can you hypothesize why so many Gaucher patients never develop PD if the link is so, like alpha-synuclein related to glucosylceramide?
That's a great question. Thanks for asking that. Yeah, I don't know. I think that there's many factors that are involved. One of the things that we haven't really talked about, that I didn't have time to talk about, was that in addition to reduced activity in glycolipid accumulation, there are other factors that are involved in driving this, at least the synuclein aggregation pathway into the aggregation pathway. Whether you have long chains versus short chains, that's one conditional thing. So we think that if a patient is somehow not accumulating long chains and they only have short chains, that they will not develop synuclein aggregates. This also matters. How in proportion of the proportion of these different structures that are occurring in the cells, whether you have the monomer really doesn't seem in our hands to be a great substrate for glycolipids, and so if this varies between patients, this equilibrium, then that could have something to do with it. We also showed that we showed in young mice, they only have this structure, the monomer structure. They don't have these physiological conformers. We're not sure why. It could have something to do with synaptic maturity, but that could also have something to do with if this is the susceptible form, maybe there's some patients that don't form this particular type of synuclein, but these are all, it's a good question. We don't know, but it's speculation.
quick question. We're
Another hard
Great
one.
talk. Were the neurons that you used, the dopaminergic neurons, were they from patients with Gaucher and Parkinson or just Gaucher, and do you think it would make a difference?
That's a good question. They were, as far as I know, Gaucher's with Parkinson's. One of the lines that we got was from Michaela Delighty and Tom Gasser's group. That was the L4P. And then some of the other lines that I didn't show that have Het mutations were from the Coriell repository and Ola Isaacson's group when he was developing those a while ago. So they did have Parkinson's disease. We haven't tested, though, which is something I'd like to do, is a line that never developed, from a patient that never developed PD.
okay. Thank you very much,
Thanks.
Joe, for a great talk.
Thank you. Should I close this out?